Firstly, thanks for the organizers for giving me this opportunity to present here today. I'm Gajender. I'm a postdoc working with Susie Hong in the Department of Psychiatry. For the next few minutes, I'm going to talk about salivary microbiome and neuroactive molecules that we are starting to explore in obesity, depression, comorbidity. As you might know, both obesity and depression are associated with chronic low-grade inflammation in peripheral tissues and in blood circulation. One of the main culprits for this chronic systemic low-grade inflammation is the pro-inflammatory cytokine tumor necrosis factor alpha, which seems to be elevated in the adipose tissues and in blood of obese individuals, and which ultimately affects the functioning of the brain. Comorbid obesity and depression are also strongly associated with cardiovascular disease, type 2 diabetes, chronic kidney disease, cancer, and several other diseases. The World Health Organization estimates that about 39% of the adult population are overweight and about 13% of the population are obese. About 5% of the world population, which is nearly 300 million people, suffer from mood disorders. So the relationship between obesity and depression seems to be bidirectional, as individuals with depression are at higher risk for developing obesity, and people with obesity have an increased risk for developing depressive symptoms. More importantly, obese patients treated for depression respond poorly to antidepressants. Although psychological stress, genetic and environmental factors, and several other factors are known to contribute to obesity and depression. And as you might know, recently the human microbiome and metabolome have been implicated in human physiology, mood, behavior, and immune regulation of the host. Here we're looking at how gut microbiome impacts the brain via multiple mechanisms. One of the mechanisms that is relevant to our study is that bacterial endotoxin lipopolysaccharide, particularly from gram-negative bacterial cell walls, may leak into the gut and cause metabolic endotoxemia, which activates immune cells and secretion of pro-inflammatory cytokines, which ultimately reach the brain and cause neuroinflammation and increase depression. Another mechanism that is relevant, I think, is gut microbiome can also synthesize neurotransmitters and short-chain fatty acids that have potential to cross not only the intestinal barrier, but also cross blood-brain barrier and affect the functioning of the brain. So far, microbiome studies in health versus disease have largely focused on the gut microbiome. And its counterpart, the oral microbiome, has been largely neglected. As you know, mouth is home to more than 700 unique bacterial species, with about 150 bacterial species per mouth, while some of these are good, some are bad, and some are ugly. And their interactions play a key role in maintenance of oral homeostasis. It is quite surprising to know about more than 60% of species that are present in the oral cavity are also residents of the gut, which suggests that the oral microbiome is important in shaping the gut microbiome. Disruption of bacterial communities in the oral cavity not only impacts the mouth, but also impacts the distal parts of the body, such as kidney, brain, heart, stomach, and also the reproductive outcomes. Therefore, we are all starting to believe that the oral systemic link is the oral microbiome. So this project, we sought to determine the composition of salivary microbiome in obesity, depression, inflammation, and in obese depression co-occurrences, and also identify individual bacterial taxa associated with specific host phenotypes. Another important goal of the study was to identify small molecules, particularly small peptides mediating microbe and microbe host interactions in these host phenotypes. So in order to address these questions, we leveraged obesity depression symptomatology cohort. We recruited about 60 participants with a wide age range between 20 and 65. We first categorized these participants into either non-obese or obese based on their BMI scores. We then categorized these participants based on Beck's depression inventory scores into either lower or higher depressive symptomatology groups. Based on obesity depression comorbidities, we further categorized these participants into either non-obese or obese with either lower or higher depressive symptomatology groups. We collected saliva from these participants and sent these saliva samples for microbiome profiling at night lab. We also sent saliva samples for mass spectrometry analysis at Dorenstein's lab. In parallel, we also collected blood from these participants and incubated them with E. coli bacterial endotoxin lipopolysaccharide. We then incubated the blood along with LPS with isoproterenol, which is a beta blocker. We then measured the percentage of tumor necrosis factor alpha positive monocytes through flow cytometry analysis, which basically gave us a inflammation control measure scores for each individual. Let me switch gears and show you some results. Here we're looking at the feature-based molecular networking analysis from the mass spectrometry data. As you can observe, we in total, we obtained about 8,000 MSMS spectra, of which only 248 MSMS spectra matched GNPS spectral libraries, which is a reference spectral library here at UCSD. I have highlighted few molecular networks that are important and that are either enriched or depleted across these 4 different groups. Here, nodes, which are small circles here, represent MSMS spectra. The edges connecting these nodes represent similarity scores. So basically, in a molecular network analysis, structurally similar molecules tend to cluster together. And as you can observe, microbial derived peptides, for instance, phenylalanine valine and tyrosine valine, which are associated with quorum sensing and virulence and are known to promote growth of beneficial bacterium, or differentially represented across these 4 groups. Another important peptide, phenylalanine dipeptide, potentially produced from Clostridium, reported to modify host cellular proteins, by targeting cathepsins in human cell proteomes. Potential dietary peptides, for instance, tyrosine, leucine, and phenylalanine and leucine. As you can see here, they're known to display antidepressant-like activity by activating serotonin, dopamine, and GABA receptors. Again, these are differentially represented here across these groups. Also, isoleucine, tyrosine, and tyrosine proline, which are known to affect dopamine and noradrenaline metabolism in mouse brain, are also differentially represented here across these groups. In addition to these, tryptophan, tyrosine, and phenylalanine aromatic amino acids are known potential biosynthetic precursors for neurotransmitters such as serotonin and dopamine, are also differentially represented across these groups. So next, we integrated microbiome and metabolomics data using neural networks. Here we are looking at potential microbe-metabolite co-occurrences. Here the arrows represent microbes and the dots represent metabolites. The directionality of the arrows represent which microbes co-occur with which microbes. An interesting finding I wanted to highlight here is Clostridia, which is reported to be a key species in several neuropsychiatric disorders and known to produce dopamine in mice, co-occurred with phenylalanine, which is a known biosynthetic precursor of dopamine, epinephrine, and tryptophan. Several
You're
peptides.
at 10 minutes right now, Gajender.
Yeah. Okay.
Gajender. Thanks.
Yeah. Done. Almost. And they all co-occurred with this. And in conclusion, or in summary, I hope I convinced you to some extent that our study provides initial evidence for microbial oral brain axis. And these micropeptide small molecule interactions hold a great clinical potential. Just another slide that moving beyond the key aspect of Kavli project is to move beyond these associations. I've been mining thousands of genomes to identify these small peptides and also assay these against human G protein coupled receptors. This will give us some targets, and also the final goal of the project is to identify effective probiotic or prebiotic or psychobiotics, basically. So with that, I would like to thank my mentors, Susie Hong, Peter Dorstein, Rob Knight, and Austin Spofford for their continuous support, and also my colleagues. And also I want to thank Kavli Institute for Brain and Mind for funding this project. With that, I'm happy to take any questions. Thanks.
Great. Thank you, Gajender. That's really intriguing stuff. So we have a couple of questions in the chat. The first one from Jen. How do dipeptides activate neurotransmitter receptors? And I think she means activate. Yeah, she says, or inhibit. Yeah.
Yeah. So it's a great question. That's what we're trying to explore here. But we still we do have some evidence from the previously published studies that these dipeptides have specific G protein coupled receptors in the enterocytes and as well as other cells in the gut that might be activated by these peptides, especially peptides with a length of between 2 and 10. Yeah.
Sorry, I was muted. So Nick has a question about the diet of the subject. Is the oral bacterial biome influenced by diet? And if so, were participants eating similar diets?
Sorry, can you repeat that question again?
Yeah, it's also in the chat if I'm not clear. Is the oral bacterial biome influenced by diet? And if so, were participants eating similar diets?
Right, it's a great question. So we haven't considered the dietary habits of these participants. This is quite a preliminary data right now, and for sure, for future studies, we must take this into the account.
I have a question for you. So when you were showing the results, it looked like you were seeing particular microbial peptides that are correlated in some ways with the measures that you made about this at the start about obesity, depression, things like that. And then you also have some speculation about relationship to diseases, like how dopamine, noradrenaline, things that would affect that might affect depression. Are the direction of those effects that you see consistent with the hypotheses that would come out of that? So for example, higher levels of peptides that would increase some of those neuromodulators, consistent with the way that drugs are thought to work with depression?
Right, it's a great question. So as you know, these are untargeted approaches that we have been using. It's basically we are trying to generate a hypothesis by data-driven approach here that we're taking. To some extent, yes, we do see some of the dipeptides like tyrosine, valine, and with these aromatic amino acids. They are pretty much consistent with the previously published studies that are either some of these are either enriched or depleted in these clinical cohorts. But some are there is some ambiguity because Right now, this is an active area of research in metabolomics. It's hard to quantify these individual peptides at higher resolution, and there's also bias with the normalization procedures right now.
There are